hantavirus is still probably very similar to that hantavirus that's causing the lung disease. Okay, so your body might recognize, whatever's causing that immunological response, it can probably recognize the same virus, that hantavirus, right? So I'm going to look for the closest answer that matches up to my reasoning. Okay. Antibody specific for the kidney disease hantavirus only? No, because I said that it's probably it's a hantavirus. It's specific to a hantavirus. That there's different types of hantaviruses. Some might cause kidney disease, others might cause lung disease. So I would not say that's the best answer. B, antibodies to the unknown pathogen, which is antigenically related to the known hantavirus. Remember, I said the immune cells in your body detect the antigens. That's what they refer to by antigenically. Those two foreign bodies contain similar antigens. So your body recognizes them as similar foreign bodies. That's why they both will be attacked. Okay? So I think that's a pretty good answer, right? Okay. C, antibodies to kidney proteins, wrong. D, the unknown pathogen. No, it's not the best answer. Okay, I would mark B and move on. Okay. In experiment 2, okay, what happened in experiment 2? He did that hybridization. Basically proves that it's the hantavirus, right? That's all experiment 2 showed. It's a hantavirus that basically thrives in your lung tissue, right? Why did the synthetic hantavirus gene sequences hybridize with nucleic acid from an infected lung? Okay, I've already discussed the answer to this, right? They reacted because the virus, the nucleic acid from the virus, it has the same DNA as the probe that we're putting in. Okay, which will target hantavirus. Okay, which is because it's a hantavirus synthetic gene. So we basically know it's a hantavirus, right? Okay. Why did the synthetic hantavirus genes hybridize with nucleic acid from infected lung? They were identical in sequence to genes from the lung. I don't know. B, they were complementary in sequence to a gene from the lung. Okay. So with hybridization, it's all that probe you put in will stick to a complementary nucleic acid. Okay. So complementary, you guys will learn that every adenine on that nucleic strand will bind to every thymine on my probe. Okay, similar, every guanine on the nucleic acid will bind to every cytosine on my probe. Okay? So they were complementary in sequence to a gene from the lung. That's probably a better answer than A, right? But let's evaluate all of them. They were complementary in structure to the RNA-encoded protein from the lung. Okay, you're never going to learn, you're never going to hear RNA-encoded protein. This has nothing to do with proteins. We're talking about nucleic acids. Wrong answer. Okay? They encoded an enzyme that bound to the nucleic acid from the lung. No. The fact that I had already discussed all these concepts, I should have marked answer B and moved on immediately. I don't even need to evaluate these other answers. Okay? So I'd put B and move on. Okay. Number 115. Why did investigators conclude that the new pathogen is a hantavirus? I've already discussed this, right? Everything that you talked about. Okay. And it has, we've talked about it has to do with everything I said. Okay. So I'll just match it up. The experiments, yeah, it shows that it's related to no antivirus. Yeah, I guess that's true. B, the experiment shows that it infects lung endothelium. Has nothing to do with hantavirus. Can also infect your kidneys, right? C, the patient data showed that the type of disease it resembled. Wait, the patient data showed that the type of disease it causes resembles that caused by no. It's a different disease, right? No pathogenic bacteria were found in the lung samples. No, nothing to do with bacteria. Answer A, right? Okay. Last question, 116, or no, 2 more questions. A principal reservoir of recognized hantaviruses in the United States and Europe is Volus. What the hell is that? Okay, VOLES, voles. I have no idea what that means. Okay, it doesn't matter. Identifying viral sources is important for control of transmission and elimination. What is the most likely source of the new pathogen? Okay, so voles is probably an animal, right?
It's like
probably a rodent or something like that, right? Okay. So identifying viral sources is important for control of transmission and elimination. What is the most likely source of the new pathogen? Okay. And I'm probably, I'm honestly just thinking of the bubonic plague. I'm just thinking back in the 15th century, what was what was one of the reasons the bubonic plague killed a third of Europe? It's because rodents were infected, ran around all the place, bit people, transmitted the disease through the biting. Okay. This disease isn't contagious through coughing or breathing, but it probably would be contagious through biting because you're getting a transfer of the pathogen through the bloodstream. Okay. So what is the most likely source of the new pathogen? I'd say a mouse. C. It's not going to be a snake. It's not going to be a fish because we're not close to fish, right? It's not going. Okay. D. A human. No, because we've already established it's not contagious from human to human. C is the best answer. Okay. 117, the investigators concluded that the new hantavirus infects lung endothelium cells. Do the data support this conclusion? We've already answered this question. Yes, it does. And we've talked about all of it. Okay. We talked about every experiment. A, yes, viral replication were demonstrated in endothelial cells. Well, viral replication is the lysogenic cell, which I talked about, which I said is probably occurring, right? Okay. So maybe that's a good answer. Let's keep going on. Yes, viral DNA was found associated with lung capillaries. Did they ever say the viral was found in the capillaries? No. So I don't think that's a good answer. Yes, viral antigens were found associated with capillary endothelium. Did they ever say the virus was found in the capillaries endothelium?
Well, in the third experiment, the antibodies bound to the capillary.
Hybridization occurred between the synthesized genes and nucleic acid from the patient's lung tissues. From the lung.
In experiment 3.
Oh, in experiment 3.
That's the antigens, or the antibodies bound to the antigens.
Okay, so the antibodies bound to capillary walls and, okay, good. So those are the antibodies, right? Okay. So that probably, but the antibodies are in our bloodstream, so they're going to be found, in the capillaries. Now, it's different from the virus making it to the capillaries. Okay, probably has something to do with the lung detects some foreign bodies. Your immune system is going to direct all your immune cells to the lungs, toward the capillaries. Okay, you understand the difference? So I would just think that, and so viral DNA was not found in lung capillaries. An immune response by experiment 3 was found, but that's different. Okay. Does that make sense? Okay. Do you disagree with me?
I mean, I would mark C.
Okay, that's fine. I might get it wrong. I've never done this passage. I've never done any of these passages. I might get it wrong too. Okay, so I'm just trying to show you the best way to go through this. And let's see. Infects organ endothelium. Okay, so what is really okay? Let's look at the other answers and then we'll pick between A and B. C. We already picked C. D. No viral antigens were found associated with lung and alveoli. No, that's wrong, right? Data shows that it does. It's there. Okay. So now we're trying to decide between A and C, right? Is that what we're deciding between? Yep. Okay. Now, so anytime I do this, I'll just go through each answer again, just mentally. Let me do this in my head. You guys come up with your own answer too. And remember, endothelial cells is kind of like the tissue surrounding the lungs. Okay, you think of endothelium as kind of like skin, but it's basically the lung cells. It's demonstrated in the cells. Okay, so hybridization of nucleic acid occurred in the lung tissue, right? And then experiment 3, they put bacteria, they took viruses, implanted these viruses into bacteria,